This is a model of the protein synthesis of the keratin protein. However, only the first 11 amino acids, or 33 nucleotide base pairs, will be modeled. The human genome codes for 54 types of keratin proteins, with them either being coded from chromosome 12 and 17. Keratin is a group of tough, fibrous proteins that form the structural framework of epithelial cells, such as the hair, skin, and nail cells. Protein synthesis begins with transcription, which occurs within the cell nucleus. It is initiated when the enzyme Rna Polymerase binds to the promoter, causing the DNA to unzip and unspiral, breaking the hydrogen bonds that hold the DNA together, separating the strand over a short length. Only one strand of the DNA contains the genetic information that is used to make the keratin protein. This is called the sense strand. And the other, unused strand, is called the antisense strand. The process of then transcribing DNA into mRNA is controlled by the RNA polymerase. The sense strand of the DNA acts as a template, and the polymerase assembles complementary RNA nucleotides and attaches them to the sense strand via hydrogen bonds, and slowly moves towards the 3' end from the 5' end, adding complementary nucleotides as it does. Length of the sense strand for keratin can vary. However, in keratin 18, it is 3,791 kilobases. The pre-mRNA molecule produced is the same as the DNA antisense strand. However, instead of thymine, it contains uracil. A RNA sugar phosphate backbone then forms with assistance from the RNA polymerase to form an RNA strand. The hydrogen bonds between the pre-RNA mRNA strand and the sense strand then break, freeing the newly transcripted pre-mRNA strand. From here, the mRNA is then edited or spliced. Splicing involves the removal of junk DNA, called introns, through the use of spliceosome, with the introns themselves coding for their own removal. Only exons are left behind to make up the mature mRNA. Within keratin 1, there are 9 exons, while in keratin 18, there are 7 exons. It is these exons which are translated into amino acids and thus expressed as proteins. The mRNA then leaves the nucleus and enters the cytoplasm via the nuclear pore complex. It is within the cytoplasm where the 3 stages of translation occurs: initiation, elongation, and termination. Beginning in the initiation stage, the mRNA then becomes associated with the ribosome in the cytoplasm, which moves from the 5' to 3' end. Ribosomes are composed of a small subunit which reads the mRNA and a large subunit which joins the amino acids to form a polypeptide chain. Then, the first tRNA molecule attaches itself to the mRNA at the start codon. This occurs when the base of the tRNA anticodon is temporarily attached to the complementary triplet of bases, or codons, on the mRNA, with an amino acid on the tail end of the tRNA. The mRNA then enters the elongation stage. This occurs when the second tRNA enters the ribosome and attaches itself to the mRNA, and then attaches its amino acid to the polypeptide chain. Once the amino acid is attached to the chain, it is then spliced up its tRNA carrier. This process continues until the ribosome reaches the stop codon. Once it reaches the stop codon, the termination stage begins. tRNA is unable to bind the stop codon. Instead, the stop codon binds to a release factor protein that causes the dissociation of the ribosome mRNA complex and allows the release of the polypeptide chain from the ribosome. Carrier proteins then transport the polypeptide chain to its functional location, where it is folded into a particular shape, determining its function. The keratin protein is an alpha helix, typical of a fibrous protein. It forms when the hydrogen bonds, linking the amino acids together, twist the polypeptide, forming a spiral, or alpha helix. This aids the function of a keratin, which is to provide strength and resilience to the hair, skin, and nail cells, as well as to allow tissues to resist damage from friction and minor trauma, as well as the regulation of cell size, cell growth, and division, as well as wound healing. If a mutation occurs during this process, research has shown that it can cause diseases which affect the skin, hair, and nails. Mutations alter the structure of keratin, which prevents them from forming an effective network of keratin intermediate filaments, causing the cells to become fragile and therefore easily damaged, making tissues less resistant to refraction and minor trauma. Even normal activities such as walking can cause skin cells to break down, resulting in the formation of painful blisters and calluses. Overall, protein synthesis is so important due to it being the making of proteins, which are responsible for all cell structure and function, something cells would not be able to live, such as exist without. This is a very complex process by which many stages are undertaken, with keratin being no different.